Just a reminder, we'll be meeting next 2 weeks from today on February 26th. And if I checked the schedule correctly, if I think I'm correct, I think we're actually going to be meeting in the Pettit building conference room instead of this room right here. But check the schedule and check the announcements that'll come out by email. And the speaker will be Mike Filler from Chemical and Biomolecular Engineering. So it's a real pleasure to have today's speaker with us, Professor Shu Jia from Biomedical Engineering. He got his Bachelor of Science in Electrical Engineering at Tsinghua University before coming to Princeton to get his PhD in Electrical Engineering with a minor in physics, and then did a postdoc in chemistry and chemical biology at Harvard before starting his academic career at Stony Brook in biomedical engineering, and then came to Georgia Tech just about 6 months ago or so, back in August of 2018, where he's currently an assistant professor in the Department of Biomedical Engineering here at Georgia Tech in Emory. He's won a couple of good awards, including NIH Mira Award and the DARPA Young Faculty Award. And so it's a real pleasure to learn what he's starting here at Georgia Tech.
Okay. Thanks, David, for the introduction. And can people hear me in the back? Good. Okay, so good afternoon. Today I'll be presenting our recent works on imaging biology across high spatial-temporal dimensions and scales. I'd like to start with a very brief introduction, just like the advent of optical microscopes that enabled remarkable how to lay the foundation for modern neuroscience. Imaging technologies have enabled new possibilities for studying and understanding biological systems. And just take the brain, for example. Till today, we tend to still agree that brain circuits still remain deeply mysterious. And this is mainly due to the lack of technologies, which really cause a knowledge gap that we can now study individual genes, molecular synapses, or neurons at very high resolution, or with relatively low resolution of the whole brain. But the challenge remains what lies in between. For a human brain, there are about 100 billion neurons and more than 100 trillion synapses. So how these molecules are integrated to support cell, tissue, and organ functions is still unknown. And to address this question, we need imaging technologies that is capable of studying molecules and cellular populations in behaving animals. And to date, there are three major challenges for optical microscopy. The first one in the spatial domain, or I call it in space, is how to perform a molecular cellular level imaging in deep tissues. And the second challenge in time, how to perform high-speed functional imaging of large-scale collective patterns of cellular activity. And the third challenge in accessibility, how to image cells in freely behaving animals. And in this talk, I'll be talking about our efforts to address all these three challenges. I'd like to start with light microscopy. In the past decades, light microscopy has enabled various biological studies at the cellular level and even at the subcellular level. And compared to other imaging modalities, optical microscopy is advantageous in its high specificity, multiple colors, as well as allowing live imaging. However, compared to modalities like electron microscopy or X-ray, the drawback of optical microscopy is its limited resolution due to the diffraction of light. For example, even if we use high numerical aperture, high magnification lens, when we focus light, the focal spot will always have a finite size, which is about 200 to 300 nanometers in X and Y, and above 500 nanometers in Z. That means any biological structure smaller than the size will become diffraction-limited blur. And also, this diffraction of light will cause this limited penetration or limited depth of focus. So to address this challenge, in the past decade, several super resolution imaging techniques have been developed, including spatially pattern illumination techniques like STED or SIM, or single molecule localization-based techniques like STORM or PALM. So in this talk, I'll be focused on STORM, stochastic optical reconstruction microscopy, which is in principle equivalent to PALM. And this is how it works. For example, this is my target structure, a cell. Instead of illuminating or exciting all the fluorophores in the cell, resulting in this diffraction-limited blur like this, our assay allows us to turn on only a small subset of the molecules. And each of these emissions comes from single molecules, which are well separated, and their locations can be determined by Gaussian fitting with nanometer precision. And then we can turn them off and turn on a different subset and repeat this process many, many times until we have enough information to reconstruct a super resolution image using all the positional information from all the molecules. Just a quick example. STORM can give you more than 10 times of improvement in spatial resolution. For example, here, we could now resolve molecular synapses in neurons, which cannot be resolved in conventional microscopy. However, as you look at these images, there are 2D images. And technically, we know technically biological samples are 3D. So how to address the limitations in 3D imaging? So for 3D super resolution and high resolution microscopy, it suffers this anisotropic 3D resolution. For example, about more than 2 times worse in Z for high NA objective. And also, there's always this trade-off between 3D resolution and depth of focus. And the depth of focus is smaller than 1 micrometer for high NA objective. So the question is, how to achieve isotropic 3D super resolution across an extended depth of focus in cells and tissues? And this is our first question. So to address this, the technique we use is optical wavefront engineering, mainly to shape the 3D propagation of waves by using digital devices like spatial light modulators and so on. And briefly speaking, the method is to encode information, X-ray information, into the incoming light so that you can extract X-ray information from the outcoming light. So optical wavefront engineering has been applied to many different types of optical imaging modalities. For example, we can use it for deep tissue imaging in the brain, or to generate pattern stimulation for optogenetics. So what we want to do is we'd like to develop wavefront engineering techniques with non-diffracting beams. Especially, there's this one type of non-diffracting beam called area beam. So as a non-spreading solution to the force-free Schrödinger equation, area beam has several unique properties compared to a standard, for example, Gaussian beam. As you can see from the simulation here, as it propagates, it experiences this constant self-bending. And also, its intensity profile remains invariant during propagation. And thirdly, it is self-healing, which means that it can recover its beam profile after being obstacled by scatterers, such as in the biological tissues. So this makes the area beam very unique for imaging purpose. So the question is, can we use this area beam to develop wavefront engineered super resolution microscopy? So we build a microscope, we implement area beam, and this is what we get. So first, we compare that with a standard Gaussian beam. And this is a Gaussian point square function taken across a 3-micron range at different depth. As you can see, above or below 500 nanometers, the point square function can be barely seen, mainly due to the diffraction of light. And this is the top view of the PSF. And this is what we got with the self-bending point square function. And we have this two-tunnel design. So as you can see, first, the area beam experienced much less diffraction, expanding by only about 2.4 times versus more than 15 times for Gaussian beam, and which immediately give us much larger imaging depth, more than 4 to 5 times of improvement in imaging depth. And also, this bending agrees very well with our theoretical model. So with this, then for any emitter, then we can always determine the position of the emitter by averaging the images in the two channels to get XY position, and also to measure the difference between the images in the two channels, and also a pre-calibration curve to get the Z position. So this is a 3D imaging technique. So with this, we demonstrate super resolution microscopy. And again, this is a low resolution image taken with the standard Gaussian point spread function. And this is a super resolution image taken with the array beam. As you can see, not only we can get much higher spatial resolution, but also different colors here represent different Z information. And we can resolve all these filaments in three-dimensional space. And this is another example imaging mitochondria in cells. Again, this is a low resolution image, and this is a super resolution image. And if we look into the screen across these dashed lines, then we can see that the hollow structure of all these, the outer membrane of mitochondria can now be very well resolved. So with this technique, we provide isotropic 3D resolution, about 25 nanometers, and image depth about 3 to 5 micrometers. And this gives us more than 2 times of improvement in Z resolution, and about 4 to 5 times of improvement in image depth compared to conventional methods. And also, we could further improve the image depth by using different engineering methods to cover a significant volume of the whole cell. And also, by using different chemical assays, it allows us to generate ultra-bright probes, which can further improve the spatial resolution down to 1 to 2 nanometers. And more importantly, the study actually allows us to generate a series of studies on non-diffracting beams or non-diffracting beam-enabled optical microscopy. One example is we know array beam has this curved structure trajectory. Then, by using this computer-addressed spatial light modulator, it's very easy just to tune or rotate the trajectory of the array beam. Then we can rotate that and get a projection of the volume of a biological sample from all different perspectives. So this is like a tomography. So using computational method, we could reconstruct the full volume of the specimen. And this is an example. We image mouse kidney tissue. And this is a volume of the tissue generated with this array beam tomography. And this is a comparison with the 10 wide field Z stacks. And they agree very well. And from the image, we can see that this array beam tomographic microscopy could give us near diffraction limit resolution at about 500 nanometers in X and Y, and about 1.3 micrometers in Z. Just to mention, for this one, we use a 40x objective lens. And also, due to the non-diffracting property, it gives us more than 10 times of improvement in depth of focus. And more importantly, for this type of tomography, you don't need any mechanical scanning. And also, the scheme can be applied to many different types of waves, such as acoustic waves or plasmonics and so on. And also, we are now trying to explore new types of physics for non-diffracting beams. And we use this new physics to generate new microscopy tools. In addition to array beam, we also study different types of non-diffracting beams, such as Bessel beams, helicon beams, and so on. And here, just a very quick example. We recently developed a Bessel beam-based imaging technique we call DRIP. So with this method, then we could improve the whole cell imaging, the depth of focus, by about 4 to 5 times. So now what we are doing, especially my student Shu Jia is doing, is we try to develop a plug-in device. Just by insert a chip into your microscope, then immediately you can get a couple times of improvement in your imaging depth. So with this, then non-diffracting beam enhance the 3D super resolution or high resolution microscopy across extended depth in cells, and also allow us to image into tissues. And to really enable tissue imaging, and there's another technical barrier we need to address. So this is the aberrations in the tissues. So just to give you a quick example is, here we have a fluorescent bead placed on top of a 10 micrometer mouse brain tissue, and we took the image. And this is the point spread function, and we see a strong coma aberration in the point spread function. And from a wave optics point of view, this is mainly due to the tissue inhomogeneity in the sample, that the wavefront coming from the emitter will be distorted, so that when they focus, they don't focus onto the same region of the camera. And this is a severe issue for imaging, especially for high NA and deep imaging, because we're going to lose the image quality, lose the photons at focus, and also because the wavefront is distorted, we're going to lose the X-ray information or the 3D imaging capability. So to address this, we ask the question, how to perform super or high resolution microscopy against tissue aberrations. Then the technique we use is called adaptive optics. So we develop wavefront engineering techniques with adaptive optics. So to do this, we add a deformer mirror into our system, and which surface can be adjusted to counter the distorted wavefront, so that after the device, we can achieve an ideal wavefront. And we combine this tool with a super resolution microscope. We call it AO Storm. And just for the same bead, this is before the correction, and this is after the AO correction. So we can see that we have much improved the point spread function brightness and shape. And more importantly, this new PSF can be used for 3D imaging. So to validate our new technique, then we imaged C. elegans. And the organ we are interested in is the microvilli on the intestinal lumen, which narrow structure is sub-diffraction limit. So it has never been visualized with optics or in intact forms. So this is a C. elegans embryo, and this is the intestinal lumen. And this is taken with a low resolution conventional microscope. And we could not resolve any structures on the lumen. And this is what we got with the AO Storm. And we started to resolve something. And if we zoom in, we can see all these actin bundles or microvilli can be very well resolved. And because this is a 3D imaging capability, so we can take a cross-section and take a look at the side view of the structure. And just to mention that this is the image of the same region without the adaptive optics. You barely resolve any structure. And if you do a Fourier transform of the image, then the peak power spectrum actually tells you the dominant periodicity of the microvilli structure, which is about 200 nanometers. And we also demonstrate this for adult worms. And we observe a very similar structure. And this is quantitative, so we can measure the size of all these microvilli. So they have this diameter of about 100 nanometers. And we can look into Z. They have this length of about 100 to 200 nanometers. And again, if we take the Fourier transform of the image, then we have this very striking hexagonal pattern of the power spectrum in the for the adult worm. And again, the peak spectrum over here actually tells us this major periodicity of about 180 nanometers. So with this AO Storm, we demonstrate for the first time the intestinal microvilli in C. elegans. And it should be mentioned that AO is also compatible with many different one-photon or nonlinear imaging modalities for cell and tissue imaging. And it can be implemented into both your illumination or detection path for deep tissue imaging. So with this, I just like to summarize very briefly that I presented our recent approaches, including super resolution microscopy, wavefront engineering with non-diffracting beams, wavefront engineering with adaptive optics to address the challenges in space. So now the next question for us is how to perform in vivo functional imaging. Or more specifically, how to perform high-speed functional imaging of large-scale collective patterns of cellular activity. And to address this, still I'd like to start with the basics about optical microscopy. for optical microscopy, it acquires this volumetric information or 3D information in a sequential scanning fashion. And as we can see, this will definitely compromise the temporal resolution. And more specifically, if we look at this simplified diagram of optical microscope, then we can see for light rays that comes to the camera sensor, it only acquires the 2D spatial intensity of light. Well, it doesn't care about from which angle these light rays come to the sensor. So the 2D angular information is overlooked. So with our new design, what we did is we insert a microlens array into the microscope so that the microlens array will refocus the incoming wavefront so that different angular information can now be redistributed onto the camera sensor. So you are capturing both this spatial information and the angular information on the same image. So specifically, we now have this 2D spatial and 2D angular information or 4D light field information. So with computational tools, we could use this 4D light field information to reconstruct the full volume information of the sample. And it should be mentioned that now you only need one camera frame to reconstruct the full volume. You don't need any scanning or a sequential image. So this means that the tool is highly scalable because the temporal resolution is only dependent on your camera speed. It's now independent from your field of view. And to reconstruct the volume, people have developed different computational tools. People first started with ray optics to reconstruct the volume. However, the angular information is recorded at the expense of spatial resolution for the ray optics model. And this actually gives us the trade-off between lateral and axial resolution. So very recently, people started to develop wave optics model for reconstruction. And by generating a 5-dimensional measurement matrix connecting the 3D volume space and a 2D camera sensor, we could use the spatially varying point square function for deconvolution to reconstruct the volume. And it mitigates the spatial angular trade-off and significantly improve the spatial resolution. So this actually enables many applications. And again, I'd like to address that this is a highly scalable technique. So the volume acquisition time is independent of the field of view. And so that it can allow for high-speed volumetric imaging with low photo damage. And people have demonstrated that in different models, including C. elegans, zebrafish, Drosophila, and mouse. It's mainly being used for functional brain imaging. And people can achieve cellular level a few microns spatial resolution with the imaging depth about tens to hundreds of micrometers, and at a fast volume rate on the order of 10 milliseconds. And also people develop different computational tools to further improve the performance. And because this is a highly scalable technique, then our question is, can we scale down? Okay, to use high numerical aperture, high magnification objective lens to image single cells and to get subcellular resolution. However, light field microscopy with subcellular level near diffraction limit 3D spatial resolution has not been explored. And there's a major challenge. It's because this wave optics model allowed the full reconstruction of densely aliased high spatial frequency through point square function deconvolution. But this aliasing is redundant near the native image plane or focal plane, and which will cause prohibitive reconstruction artifacts. So I'm not sure if you can see this clearly, but this is just to show there's a strong artifact near the focal plane. So this is prohibitive, and people have to image on only one side of the focal plane or use coded wavefront. But these methods compromise image depth or resolution. Or people could combine with advanced algorithm, but they're mainly designed for functional imaging, calcium imaging. So these methods so far are not impractical for high resolution microscopy. So, and more specifically, this is a conventional setup for light field microscopy. And there's another setup called planar optical camera 2.0, where this microlens array forms an imaging relationship between the focal plane and the camera sensor. However, for this design, it compromised the volumetric imaging capability. So what we come up is somewhere in between, where this microlens array forms this defocused imaging relationship between the imaging plane and the camera sensor, where this distance A can displace the artifact region by A over M squared. M is the magnification of the system. And B is the can control the optimum dense sampling of both spatial and angular information. And this is the typical parameters we use for the design. So with this, and in our numerical study, we can see that this is a conventional light field microscopy near the native imaging plane, where you can see this strong aberration, strong redundant aliasing area that cause the artifacts. And this is our new design, where this artifact region can be totally avoided. So with this, we first tested with the fluorescent beads. And this is with the conventional light field microscopy. As you can see in the reconstruction, that this 3D reconstruction can now give you a faithful 3D information of the bead cluster. Well, with the new design, we can obtain this 3D reconstruction of the bead cluster very well. And we first imaged fixed cells. In HeLa cells, we imaged mitochondria. And this is an image taken with conventional light field microscopy, where you can see there are strong artifacts, and also there are poor resolution, especially in Z. So the actual information is actually lost. So with high resolution light field microscopy, now what we can see is, if we zoom in, we can compare. We gain this volumetric information again. And oh, here I need to mention different color in all these images represent different Z information. And also, we can see that the artifacts is significantly reduced. And also, we have much improved the resolution. And here, this is an image with a wide field microscopy. And we can see this is a 2D technique without any 3D information. So with this, we started to we calibrate, and we determine the resolution is about 300 to 400 nanometers in X and Y, and about between 4 to 1,000 nanometers in Z. And also, the image depth is able to cover a significant volume of the whole cell, giving about 5 times of improvement over conventional methods. And it should be mentioned that here, in the axial dimension, we achieve a 400 nanometer axial resolution, which is slightly below the diffraction limit. And we reason that this is due to the optical sectioning by the microlens array. So with this, we started a live cell. And this is the image for mitochondria in HeLa cells. So just to mention that different color, again, represent different Z information. And just for single frame, this volumetric data is generated from only one camera frame. And because this is a quantitative method, so we can always zoom in and follow the dynamics of the organelles in the cells. And the technique actually gives us a tool to measure cellular structure as well as the dynamics in a volume context. And then we accelerate the speed to image Golgi-derived membrane vesicles at 100 to 200 hertz. Then we found that we can successfully track all these vesicles in the cells with high resolution. So it should be mentioned that this high-resolution light field microscopy provides a high-speed, whole cell level imaging tool for answering previously inaccessible questions. And this is mainly due to, for example, confocal or two-photon microscopy. They cannot achieve such a temporal resolution across the same field of view. And just to test how fast we can go, we image fast-moving particles at 1 millisecond. And we can track successfully these particles in 3 dimensions. And we can reconstruct the particles and use Gaussian fading to track their positions. So this allows us to demonstrate this nanoscale precision particle tracking at ultrafast speed across a 3D volume in this case. Okay, so with this high-resolution light field microscopy, we're able to demonstrate near diffraction limit spatial resolution and large imaging depth, and also fast imaging speed. And also, it should be mentioned that the technique is fully compatible with super-resolution microscopy. That means that a combination of the two techniques will give us both the spatial resolution and the temporal resolution we need to address structural or functional information in the cells. And also, very recently, we found out that if we image instead of the real space but in the Fourier domain, then when we reconstruct, we are able to fundamentally remove all the artifacts. So we developed this technique called Fourier domain light field microscopy. So just a quick overview of this Fourier domain microscopy is we have no reconstruction artifacts, and also we have significantly improved the imaging depth. We obtain 80 micrometers versus 20 to 30 micrometers in the conventional techniques. And also, the spatial resolution can be improved to 1 micrometer using the 40x objective lens. And also, it should be mentioned that the computational cost, because here, instead of using the 5D measurement matrix for the deconvolution, now we can use a unified PSF in the Fourier domain. So just to use a basic desktop computer, we can achieve seconds reconstruction time versus hours using with the previous method. And this allows us to further develop a technique for real-time functional imaging. And also, now my lab is working on to develop the next generation light field microscopy for imaging neural activities and single cells. And also, we try to apply that to different modalities. And also, in addition to experimental tools, we also develop imaging in painting techniques just to improve the imaging speed. And the technique is mainly to use sparsely taken data and to in-paint useful information so that we can obtain a full resolution of the image. And for example, here, it allows us to reconstruct this actin structure using only 1% of the original data. And also, we develop machine learning tools to generate example libraries that can convert low-resolution image into super-resolution image. And this is experimental results showing microtubules, and this is the experimental ground truth. This is the low resolution. Okay, so with this, we also studied into the camera physics for imaging. This is mainly because for CMOS camera, this is getting more and more popular for bioimaging due to their large field of view and high speed. So, however, the noise is a problem fundamental for the technique for the CMOS camera sensor. So we try to address the noise in the sensor and develop a tool called automatic correction for CMOS-related noise. And we demonstrate this by accessing and eliminating the noise in the sensor to achieve the performance of an ideal camera. And we demonstrate in multiple imaging modalities, so to prove the usability of the technique. So with this, I presented high-resolution light field microscopy, Fourier domain light field microscopy, computational microscopy, and our recent studies on camera physics to address challenges in time. So here come to the next question. Then, how to image live animals? Or more specifically, how to image cells of the functioning brain in freely behaving animals. And the motivation is mainly that because the microscope like this is not compatible with animal like this. Okay, so to do that, nowadays people have like different techniques. So one is to image on awake height-restrained 2-photon imaging platform. And the tool offers high resolution, motion correction, and also it's fully compatible with artificial environment or VR. And another category is to develop miniaturized microscopy. And this miniaturized microscope actually gives us wide field imaging, faster imaging speed, and also this is compatible with rodent behavioral assays. And also, this can be implanted onto the mouse brain, so it allows longer time-lapse experiments. So with this, now what we want to do is to identify the mapping between the brain function and the structure. So the first step is we try to develop super-resolution miniscope. And we first developed the hardware and software of the system, and also the parameters for the miniscope agree very well with the commercial products, and it's very light and provides cellular level resolution. And one way to do that is through computation. So the method we develop is based on the gradient analysis of the points per function. And we use that to reduce the size of a points per function. So with that, we first test on the conventional microscope. So this is a conventional image of microtubules. And this is what we call double-grass image of microtubules. And this is experimental ground truth. And if we zoom in, we can see it already gave us improvement in the spatial resolution. And then we tested on a miniscope. So first with fluorescent beads. So this is the conventional image. What we can see is it's blurry and aberration, and also we can't resolve much. And with the double grass, then we can resolve the beads within these blurry regions. And if we measure that, then we can see it can resolve many of the beads that could not be resolved with conventional methods. So this can give us several times of improvement in resolution. But for the major application of miniscope, the main challenge is the tissue imaging, which has this high background, overlapping signals, and aberrations. So with this, we developed another method called the BSSE to address this challenge. So I'll just skip the algorithm over here by showing you the results. So here we first tested BSSE with brain tumor tissues. And this is the raw data. As you can see, it's blurry. And this is with BSSE. And if we zoom in, we can see that now the cellular structure can be much better resolved. And if we take some measurement, we can see that it provides cellular level resolution and also provides us new information about the cells in the brain. And also we tested with the mouse kidney tissue. So this is the kidney cortex. And we can see it's, the image is blurred by this high background and also overlapping this tubular structure in the kidney. And this is the BSSE processed image. And if we overlay these two images, then we can see that the method mainly enhances all the signals while suppressing the backgrounds. So now we have this signal enhancement, background subtraction, and also cellular resolution. And we also tested with calcium imaging. So this is the raw data, and this is the BSSE process data of the neurons, of the neuron firings. And also this is another latest technique we use as a ground truth for validation. So, and then if we process the data and we zoom in some of the regions, then we can see that this is the top row is the raw data, and this is the BSSE data, and this is the ground truth. We can see that the method could give us could help us obtain previously missing information, for example over here, or could help us reduce false positives in the tracing. So actually, this enables us to not only perform structural imaging using the miniscope, but also the functional imaging. And hopefully, we can combine the two. And this is an ongoing research. So we have this resolution improvement, and we like to implement more colors and also 3D imaging capability, and also to apply this to many different applications, not just the brain, but it could be other organs. And also another thing of interest is because this is so flexible, then instead of using one, then we can use an array of them, right? So, and because this is so small and compared to a big microscope, it allows us to attach it to our biological sample and image in its native culture in the incubator. So this is what we recently developed. We call this system MEM, and so we try to develop an array of these miniscopes, and then so that they can image different wells at the same time in the incubator for long-term imaging. So this is still ongoing, but here I just want to address that miniaturized microscopy provides us the accessibility and also opens up many new possibilities for research. So with this, I'd like to conclude. I hope I've presented our recent techniques to address challenges in space with super-resolution microscopy, non-diffracting beams, adaptive optics, high-resolution light field microscopy, Fourier domain light field microscopy, computational microscopy, and the new camera physics, and also this miniscope, and also the tools to improve the resolution, and also to enhance the throughput. So with this, I'd like to thank first my lab members. Without them, the work wouldn't be done. And also, I'd like to thank all my former members and collaborators. Thank for their valuable contributions. And finally, I'd like to thank the sponsors for our research. And lastly, if you have any further questions, feel free to talk to me or any of my lab members. Thank you.
Questions? All right, I will start. So do all of these techniques or techniques that you showed use added probes, or do any of them use endogenous fluorophores or chromophores for the imaging? Or are they all added?
Yeah, most of them are fluorescence. Yeah, just with labels. Right.
Is there anything that can use kind of natural?
Yeah. A lot of techniques are compatible with, for example, label-free or intrinsic signals in the sample. Right. So just for example, the miniscope, it can image intrinsic signals and also phase information from the sample.
For the high-speed, high-res imaging, you're pulling out the X, Y, and Z coordinates. Are you using a second camera to triangulate to get the Z, or how are you getting the Z in?
You mean for the light field microscopy?
Yes.
So we only use one camera, and the good thing for this technique is because you are using microlens array, it will redistribute the light information into different regions of the camera, right? So for one image, it's not just the spatial information. You also have the angular information, and you need an algorithm to reconstruct the original volume.
If not, let's thank you one more
Thank you.
time.